In the last class, we have learnt about spermatogenesis. In this class, we are going to discuss about oogenesis, human reproduction, part 5. What is oogenesis? Oo means ovum. Genesis means formation. It is the process of development of female gamete, namely ovum, from the ovaries of female reproductive system. There are various stages in the development of ovum. They are formation of oogonia, formation of primary oocyte, formation of secondary oocyte, and formation of ovum. The first stage in oogenesis is the formation of oogonia. Oogenesis starts in the embryo at the 7th week of gestation or pregnancy. At that time, the primordial germ cells or undifferentiated stem cells from the epiblast of the embryo move towards the newly formed ovary, divides by mitosis, and forms millions of oogonia. Oogonia is otherwise called as egg mother cells. These oogonia are diploid in nature. At the same time, the germinal epithelium that surrounds the ovary produces bud-like cells called follicles. The formation of follicles is called folliculogenesis. Now, the follicles surrounds a single oogonium and develops into primordial follicles. The main function of the follicles is to support the oogonia. The next stage in oogenesis is the formation of primary oocyte. Now, the oogonia formed from the primordial germ cells undergo mitosis and form two diploid primary oocytes. Now, the primary oocytes again undergoes meiotic division 1 for replicating their DNA, and the division stops at the prophase 1 of meiotic division 1. This meiotic division occurs pre-natally, that is, before birth or during pregnancy. Then, the primary oocyte is surrounded by a single layer of granulosa cells to form primary follicles. As large number of primary follicles degenerates from birth to puberty, only 60 to 80,000 follicles remains in each ovary at the time of puberty. Then the primary follicles is surrounded by many layers of granulosa cells and it develops a single theca layer to form secondary follicle. Now, the secondary follicle develops a fluid-filled cavity called antrum and changes into a tertiary follicle. In this stage, the theca differentiates into two layers, theca externa and theca interna. The next stage is the formation of secondary oocyte. When the female reaches the age of puberty, the hypothalamus of the brain releases GnRH, that is gonadotropin releasing hormone. This hormone in turn stimulates the anterior pituitary to release FSH and LH to the ovary for the first time. This removes the block in the meiotic division 1 and causes the growth of primary oocyte inside the tertiary follicle and to complete its first meiotic division. By this process, the primary oocyte gets converted into secondary oocyte and a first polar body. Now, the polar body undergoes meiosis to form two second polar bodies. If there is no fertilization, the second meiotic division never completes and the egg degenerates. Now, the unfertilized egg is eliminated from the body along with the lining of the uterus during menstruation. Finally, one haploid ovum and 3 polar bodies are produced at the end of oogenesis. Polar bodies or haploid cells form due to the unuchal division of the oocyte. This unuchal division results in the formation of larger cell with more amount of cytoplasm and a smaller cell with less amount of cytoplasm. The cell with large amount of cytoplasm develops into ovum and less amount of cytoplasm becomes the polar body. Even though polar bodies have the same amount of DNA as that of the ovum, as they lack cytoplasm and other cell components, they do not have the ability to fertilize with a sperm and to develop into zygote. These polar bodies helps to remove one half of the diploid chromosome set produced by the meiotic division in the egg. The final stage in oogenesis is the formation of the ovum. If there is fertilization or after the penetration of sperm, the secondary oocyte undergoes second meiotic division and form a ovum and a second polar body. The second polar body also degenerates. Now the tertiary follicle becomes the mature follicle called graffian follicle. Now let us see the structure of human ovum. As human ovum is not covered by a shell, it is said to be non-cleidoic. As it contains no yolk, they belong to alecithal eggs and they are microscopic in nature. The cytoplasm of the egg is called ooplasm and it contains a large nucleus called germinal vesicles. It is surrounded by 3 layers or 3 coverings. The inner membrane is thin called vitelline membrane. The middle membrane is thick called zona pellucida. The outer membrane is thick called corona radiata. The vitelline membrane consists of protein fibers with protein receptors that helps to bind with the sperm during fertilization. Zona pellucida involved in communication between oocytes and follicles during oogenesis, protecting the oocytes, preventing polyspermy, etc. Corona radiata is a glycoprotein layer that supplies vital proteins to the egg cell. Between the vitelline membrane and the zona pellucida, there is a narrow space called perivitelline space in which the cortical granules released from the ovum gets deposited. In this class, we have learned what is oogenesis and the various stages of oogenesis, what is primary follicle, secondary follicle, tertiary follicle, and the structure of human ovum. Meet you in the next class with another video.